Hello everyone, we are studying genetics. We are studying in 12th class. In 12th class genetics, we are studying the second chapter of genetics, that is called molecular basis of inheritance. Okay, molecular basis of inheritance term we had elaborated this, and today the subtopic of molecular basis of inheritance, that is the gene regulation in eukaryote. Okay, so term here is the gene. So what does meaning gene has? That suppose this is our DNA or chromosome. Okay, so some part of DNA, there is some part of DNA. DNA means DNA contains some part. Okay, some part in DNA is functional, and that is called gene. So what is the meaning of gene? Functional part of DNA. Functional part of DNA. And we had studied that DNA having 2 or 3 parts, that is replication, transcription and mutation. Yeah, there is only 3 work assigned to DNA. So here we are getting the transcription, means gene regulation. We are studying here the transcription, functional part, that is the transcription, means from DNA it will form the mRNA. Okay, now what will happen? It will form the mRNA, and mRNA, from mRNA we will have the protein. Okay, so functional part of DNA, means gene. Gene is the part of, we can write that gene is the part of DNA. Gene is the part of DNA which forms the protein. Okay, this is the meaning of gene. So now we know that gene. Okay, then another term of subtopic, we are having regulation. Another term is the regulation. So regulator, we are doing very regulator. What we are doing with regulator? What we are playing with regulator? Regulator, we are keeping the speed of fan either high or low. Means this regulator is the part. Regulator is the part on which we can control the speed of fan. Okay, so process is the regulation, and regulator is the suppose can say the technique, or so here we are having the regulation, means the speed of expression of DNA, speed of expression of gene is getting regulated, means protein formation will getting up or down. Okay, up and down, that is the gene regulation. So today's topic is gene regulation in eukaryote. Okay, we will see the gene regulation in eukaryote. Okay, you come with me and see what will happen. So this is the eukaryotic cell. This is the eukaryotic cell, means can say this is the nucleus. Eukaryote means it will have the nucleus. Karyote means nucleus. So nucleus is having the double membrane. So you can say the double membrane and discontinuous nuclear membrane, and there is the discontinuous part which we are saying it nuclear. Okay, this is the diagram of nucleus. And around the nucleus there is another membrane, that is called cell membrane. Okay, look here. So it is the DNA. Okay, double stranded DNA. So DNA we are having 3 to 5 and 5 to 3. We are knowing this. Okay. So in the DNA also there is a part called, we have gone through all this. We have gone through all this in a splicing. Okay. So in transcription, what will happen if in case of mRNA formation, what will happen? Means one strand, that is 3 to 5, if it get translated, means get transcribed or get encoded into mRNA, that is hnmRNA, is called 5 to 3. Okay, and this mRNA, this strand is acts as template, and we will have this fragment. That is, we are going gene in, we are calling it gene in DNA. Okay, so here we are telling this exon and intron. This black color is intron. So what will happen again in case of eukaryote? There will be process which we are saying it. There will be process which we are saying this is splicing. So a splicing, what will happen? Though there is a two exon, these exon will comes together and intron will be split out. That we are saying this splicing. So how many process will occur in nucleus? So nucleus we are getting transcription and a splicing. So this is now functional mRNA. Okay, where functional mRNA get formed. So functional mRNA formation is taking place in nucleus. Okay, now till first step, second step, these mRNA will comes out to cytoplasm, comes into cytoplasm from nucleus. This is the third step. And fourth step, this mRNA ribosome will bind and it will form the protein. So it will form the protein. So protein formation means translation is the fourth step. So there is a fourth step in case of eukaryote where protein formation there will be up and down. Protein formation, speed of protein formation will up or down. So gene regulation in case of eukaryote you can control at fourth step. Yeah. Another more important question you can ask with yourself. So I will help you to opt. Yeah, there will be questions at gene regulation in prokaryote. Okay. So you are knowing in case of prokaryote there is no nuclear membrane. So transport there will be no transport and there will be no splicing. So how many steps will be? Transcription and translation only. Okay, this is the gene regulation. Okay, then another topic after this page. Another topic of after this page is called Operon concept. Okay, this is called Operon concept. Operon concept was given by Jacob and Monod. Yeah, first question is that what is the operon? Operon is the fragment of DNA. Operon is the fragment of DNA that contain structural gene. Yeah, that contain a structural gene, operator, promoter, regulatory gene and promoter of regulatory gene. So this fragment of DNA contain a structural gene, operator, promoter. Okay, operator, promoter. So there is no need. It is not necessary that control or regulator gene will be present like this. So there is a regulator gene which controls the expression of a structural gene. Then there is a promoter. Promoter we have studied. Remember. So there is a promoter which controls the structural gene. This fragment is called operon. Okay. Operon. Operon is the fragment. What we can write now? You have to note it down. Operon is the fragment of DNA which contains the structural gene, operator, promoter, regulator gene and promoter of regulator gene. Okay. So operon is the two type. Operon is the two type. Okay. Operon is the two type. One is called inducible operon. Another is called repressible operon. Okay. So now you have to know what is the inducible and what is the repressible. By example, you can study this. Okay. I will try to help you out with the definition with the example. Suppose now fan is stopped. So there is no process going on. A fan is not working. Okay. So what we will do? Switch will on. We will keep the switch on. Then fan will rotate. Start rotating. So yeah. Now you think what will happen? So one condition, fan was not working, there is no expression of gene was taking place, then some external factor will come and switch it on. The function of gene will be regulated, that is inducible. Means operation system was not working and we are keeping it to work. That is the inducible. Another thing is the repressible. Repressible means system is working, fan is running. Yeah, another example you think, same example, two side. Fan is running, means system is working and we are switching it off, means stopping it. That is the repression. Okay. The example of inducible operon is lag operon. Lag operon. Lag for lactose. And another example, that is Operon. Operon. Trip for tryptophan. Okay? Trip for tryptophan. So in NCERT, there is no system to study. It has not mentioned for the repressible operon. So we will study the inducible operon only. That is lag operon. What the operon is? In a lag operon, we are having the three structural gene ZYA sequence. You must remember, sequence should not be reversed, sequence should not be shuffled. It is ZYA. Okay. So we have studied. So there will be operator, then there will be promoter, then regulatory gene which control the expression of a structural gene, and promoter. This is the operon of operon. This is the operon model called lag operon. Okay. So now there will be two conditions. Okay. In lag operon there will be two conditions. One condition, there will be absence of lactose. Lactose is absent in medium. What lactose is? You are knowing lactose is the disaccharide. It is made up of glucose and galactose. Glucose and galactose after getting polymerized, after linkage formation between the glucose and galactose is formed the one molecule of lactose. So lactose is absent in medium. What will happen? This is the inducer gene. Inducer gene will form gene, means it will form the mRNA, and mRNA will form a protein. mRNA will form the protein. This is called repressor protein. Okay, this is called repressor protein. What called? Repressor. Repressor means it will repress the system. What it will be do? It will bind to operator. Okay, it will bind to operator. Now you are knowing that polymerase will bind to promoter. Okay, polymerase will bind to promoter. And what polymerase are doing? They are encoding the language written in this structural gene. Means helps in formation of mRNA. RNA polymerase helps in formation of mRNA. So what will happen if now polymerase will progress, when polymerase will try to progress towards structural gene, so there is a hindrance. There is a hindrance will stop the progress. Means that is, so this condition, there will be no mRNA formation, and this condition is called switched off. This condition is called switched off. Means there will be no mRNA formation will take place when lactose will be absent in medium. Okay, this is the first condition. And another condition in same diagram you can see when lactose will be present in medium. Okay, When another condition will be lactose will be present in medium, what will happen in that case? We will see here. Okay, lactose will be present in medium. So when lactose will be present in medium, what will happen? You can realize. So it is the repressor protein as usual. It will form the mRNA, and mRNA will form after translation. It will form the repressor protein as usual it was doing earlier. Okay, but lactose is present in medium. Lactose is having affinity, comes and bind to repressor protein. Okay, it will bind to repressor protein. Now what will happen? As usual, it will try to go and bind on operator. Okay, it will repressor protein will try to go and bind with operator, but it will not bind. Why will it not bind? There is a docking site of a specified diameter, but after binding this glucose lactose, its diameter will increase, so it will not bind. Okay, first condition you have to note down. Then another condition, what was happening? Polymerase will bind to promoter. Now when polymerase will bind, progress toward 3' end, it will progress towards this, and it will encode the all 3 gene structural gene, and it forms this protein or enzyme. Can say that beta galactosidase permease. You have to remember is that which gene, what type of protein it's formed from which structural gene. And here is the transacetylase. So what is getting happen here? When lactose, and this condition is called switched on. This condition is called switched on. So what two conditions we have seen? The two conditions. In one condition, lactose was absent, so there is no structural gene was unable to form the protein or enzyme. When lactose will be present, there will be protein formation or enzyme formation taking place. This condition is called switched on. Okay, this condition is called switched on. So we have seen the two conditions. Yeah, another more important one question is coming. The lactose bind to repressor protein is called. This is called allolactose. Okay, you must remember. This is called allolactose. Lactose, the types of lactose which binds to repressor protein is called allolactose. So now I am concluding this. So another thing is that another important thing is here is asking the negative and positive regulation of lag Operon. How many factors you can remember? How many factors was promoting the synthesis? How many factors means you can say you are promoting the synthesis, that was the polymerase. Okay, polymerase presence is important. So polymerase. What was the factor was the inhibiting? That was the lactose. Lactose was inhibiting. Okay, glucose was inhibiting. That your question is present in exercise of NCERT. Another more important, what was the lactose diagram is given in NCERT. Below that, a question is given that both are same question and same answer. Okay, thank you. Today's topic we have studied. Thank you.